So last week, we looked at Introduction to Biology and Microbiology. So from last week's study, we looked at also the origin of living things. First of all, what is biology? What is microbiology? We define biology as a study of life, where bios means life and logos meaning study. And also we looked at microbiology as the biology that looks at what microscopic organism. That is, organisms that cannot be seen with the naked eye. Okay, so we also learned about the origin of living things, and we talked about several theories, and we looked at their pros and cons, the ones that were rejected and the ones that were accepted. We made mention of the spontaneous generation, whereby it was believed that at the beginning of life, everything just existed and people started living. And I mentioned that that theory was what disproved. And the current theory being used now is the what modern theory. It states that life originated from simple organic compounds. Now, this modern theory was what laid upon theories upon experiments conducted by two individuals, Miller and Urey. He stated that these two stimulated what early earth condition to demonstrate that life can be what produced. Sorry, to demonstrate that amino acid can be produced from what normal earth minerals. And he made mention of scientific method. I was telling us last week Saturday that scientists, they don't just come out and start giving us laws and theories. And there are specific methods that do what guide their conduct. So we looked at the step-by-step approach to what establishing a scientific theory. We said number one was what observation. Number two, questions. Number three, generation of hypotheses. Then you experiment, your data collection, then your conclusion. Okay, so fast forward to what we have today. We'll be looking at cell biology and statistics. Cell biology and biostatistics. All of us can see the slide. Okay, session objective. Number one, after the end of this class, we should be able to explain the origin and characteristics of the first cell. Two, to be able to differentiate between prokaryotic cells and eukaryotic cells. So you have to know first of all what is prokaryotic cells and then what is eukaryotic cells. Then you'll be able to also differentiate between the two. Thirdly, you understand what the cell theory, not origin of life, not theory of life, the cell theory. 4, you also be given a brief introduction to basic biostatistics. 5, you also be taught on how to use a microscope. So, origin of first cells. Now, first of all, from last week, we understood that cells life originated from non-living things. Now, this is called abiogenesis. It's a hypothesis explaining the natural origin of life from non-living matter. Now, from last week, we also established the fact that this theory was founded upon experiment by two individuals, Urey and Miller and Urey. We say that these people, first of all, they stimulated the early earth condition. That's, they were in the laboratory. They tried to create a scenario that resembled the earliest form we had in earth to prove to us that life can arise from non-living matter. So we'll be looking at the chemical evolution. One, now, formation of simple organic molecules. Now, around the earliest time, the Earth is theorized to be billions of years old. Now, the Earth has this very harsh environment with a reducing atmosphere. It was composed mainly of gases like methane, ammonia, hydrogen, water vapor, and energy sources, lightning, UV radiation, and volcanic activities. So this atmosphere, the gases, the lightning, and everything, there was a series of reactions. Now, the reactions produced simple organic molecules called the amino acid. Hope you are still together. Now, We see that at the earliest time of Earth, that there were numerous gases, mention of methane and ammonia. Now, these gases were in the atmosphere, and then there were also what sources of energy. You know, wherever there is what energy, there is likely to be life. So the atmosphere contains gases and chemicals, and then the energy source, the lightning. Now, this induced what a reaction, series of reaction, and eventually it led to the rise of what organic molecules. Now, these organic molecules what they became the what building block of what life. Okay, so now what we want to actually what we want to look at is how did this life what evolve. So we have now established that life began what from non-living things. Now it began from what series of chemical evolution, series of chemical events that led to the formation of simple organic molecules. How did these simple organic molecules come to be? They come to be by what reactions that what occurred during the earliest form of Earth, during the earliest time. These reactions were due to what out of lightning and gases. The lightning provided what sources of energy, and then the gases provided what minerals needed. So these chemical elements they reacted together, leading to rise of what simple organic molecules, which are amino acid and nucleotide. Now, last week I told us that there was four basic macromolecules for life. Number one being proteins, number two carbohydrates, number three lipids, and number four nucleotide. Now, these chemical reactions led to the rise of what? The first organic molecule, which is amino acid, and also to nucleotide. Now, second point there is polymerization into macromolecules. Poly means multiple. Merization means different parts, that are smaller parts. So polymerization means what? The coming together of these little molecules to form what? A very large macromolecule, which is the protein. So now, first of all, I told us that at the earliest time, they were just gases, they were lightning. These gases and lightning formed a reaction to produce what? Very simple organic molecules called the amino acid. Now, these amino acids, they started what? Coming together. They started fusing, they started reacting with each other, and before you know it, they formed an aggregate to produce complex macromolecules. Let me go again. The amino acid, there are smaller molecules born from what? Normal non-organic components like methane gas and just lightning. Now, these non-organic components form the what? Organic molecules, which are very small. These organic molecules now come together to merge and form a very big macromolecule. Now, the simple molecules combine to form complex macromolecules like proteins and RNA through what is called condensation reactions. Please, if you are writing, you should take some things down. So, first point there is formation of simple organic molecules. Secondly, polymerization into macromolecules. Whereas what happened there is that these simple molecules formed initially, they combine together to form complex macromolecules. Are you following? Hope you understand. Okay. Please, at any point, if you are lost, just unmute your mic and ask your question. So now, when these macromolecules are formed, they also what undergo what polymerization. They keep what adding together and interacting with another to form the earliest type of cell called protocells. Called the what? Protocells. Protocells. Proto simply means earliest or primitive or primary. Then cell simply means what? Chambers. So these polymerizations into macromolecules underwent so many aggregation of macromolecules. Proteins came together, amino acid came together to form proteins. The proteins came together with carbohydrates, nucleotide, and other ones to form cell-like structures. Now, and these cell-like structures eventually would aggregate to do what? To form the cell which we know of today. Hope you are still together. And let's recap of everything again. Let's recap. The origin of first cells. Now, the earliest form of cells originated through a process called abiogenesis. Abiogenesis simply means was origin of life from non-living matter. So how did life come to be from non-living matter? First of all, there was a series of chemical evolution. And this chemical evolution was, we broke it down into three steps. Number one, the first step there was formation of simple organic molecules. Second step was what? Polymerization of these simple organic molecules to now form macromolecules. Macro simply means large or big. So the simple molecules came together to form the macromolecules. The simple molecules came together to form the what? The macromolecules. Now, is there any question so far? You can also use the chat in case there is anything you are missing. So there are key experiments that support this idea. One of these experiments includes what? The Miller-Urey experiments. Now, these young women are the ones that was stimulated the earliest earth condition to show us that life can what? Generate from non-living matter. So moving to the next slide now. The earliest cells. So the earliest cells, one of the earliest cells that was formed was called the stromatolites. So what is stromatolites? There are simply what the layered sedimentary formations created by microbial communities. Now, let me explain. Stroma simply means layer. It's a Greek word meaning layer. Whenever you hear stroma, it communicates of what layers. The light, L-I-T-S, simply means what? Stone. So stroma light simply means what? Is a kind of like term to explain layers of stones. Now, the earliest cells formed were what? Microbial communities. These earliest cells came together what? And started stacking stones together, kind of like sediment particles together to form what? Layers. So this is what the proof of the earliest cell. Now, let me recap it. Let me go again. Stromatolites are simply what? Layer sedimentary formations by microbial communities. This had what? A very special role in shaping the Earth's environment. Also, looking at the microfossils, they are what? Remains of microscopic organisms. The cyanobacteria, and then led to the great oxidation environment and the evolution of complexity. Are we still together? Are we still together? All right, let's go over this one more time. So we want to look at the evidence of the earliest cells. So how did these earliest cells came to be? Now, we are saying that, okay, first of all, there were no human beings during the formation of Earth. So basically, everything we are doing is more like speculations. But why this is not speculation is that every single thing we do is backed by proof and by data. So we started asking questions. How can we know that cells originated? Then we proved it before that cell originated from non-living materials into what? We proved it before that cells originated from simple organic molecules into macromolecules and then protein cells. And now we want to look at evidences that supported what? The earliest cells. As we want to trace the complex cell we have now to the earliest beginning. Now, looking back, we see there is something called stromatolites. We found out that we are what? Layered of sedimentary formations. These were very organized and cannot be formed by normal, it cannot be just formed by spontaneous something. Now, when scientists looked at this, they discovered that these sedimentary formations were created by microbes, that's microorganisms, primarily what? The cyanobacteria. It dates back to as far as around 3.5 billion years ago, making them one of the oldest evidence of life on Earth. So these stromatolites prove the fact that what there were lives on Earth as far back as 3.5 billion years ago. Now, How did these stromatolites came to be? What was the formation process? Now, microbes, that's microorganisms. Whenever I hear microbes, I simply mean microorganisms. Now, these microorganisms, they were possibly most of them were photosynthetic bacteria, just like cyanobacteria. Now, they come together and then they trap sediment particles, small sand-like material, and then secreted calcium carbonate. So these microbes, they trap sediment particles and secrete calcium carbonate into it, kind of like your super glue and your sand. And before you know it, they are forming layered structures over time. So these stromatolites now what serves as a very concrete proof and evidence that what there was life. It, someone's mic is on. Do you have a question? Hello? Okay, so your mic is on. Do you have a question? Okay, so you can continue now. So now we have looked at the first proof of the earliest life on cells. Now we'll be going further toward other proofs. Now, the second concrete evidence that there were life on Earth as far back as 3.5 million years was the presence of microfossils. Now, what is this? What does this mean? This simply means when you hear about fossil fuels, it simply means remains of microscopic organisms. So the remains of these organisms, probably a whole entire race of microscopic organisms, their remains, when they die, their remains are preserved in ancient rocks. So these remains, when extracted from these rocks and studied, they help us date back life as old as that rock is. So they provided direct evidence of early life forms. And they also reveal the morphology and signs of ancient microbial life. Okay. So the examples, now there's this very popular example. Let me try to remember. Now, there was this one found in Western Australia. It dates back to around 3.4 billion years. That there is a fossil microbe found in rocks that's around in the western part of Australia. Now, when these fossils were extracted, they dated back as 3.4 billion. Okay, so the third concrete evidence as well is what cyanobacteria. So this is the first primary cell that kind of like was verified. The cyanobacteria. So what are cyanobacteria? Cyanobacteria are a type of photosynthetic bacteria. They played a very important role in what oxygenic photosynthesis. Hello, please. If you have a question, can you help me unmute your mic? Okay, so let's continue again. I was telling us that the cyanobacteria, they are the first organism to perform oxygenic photosynthesis using sunlight to convert carbon dioxide and water into glucose and oxygen. Now, first of all, what is photosynthesis? Primary school question. So photosynthesis is what? The process by which plants make their own foods through the help of sunlight and water. So cyanobacteria are the earliest organisms. They use photosynthesis to generate their own food, and also very important to release oxygen into the environment. Remember, when I was telling us that the earliest form of Earth, they have numerous gases, methane, and the rest. Now, when the cyanobacteria came into play, they started synthesizing these gases using the help of sunlight to produce energy, to produce oxygen.
Sir, please, your voice is not audible enough. I can't hear you very well, and I'm not seeing your screen, Sev.
Are you seeing my screen now?
No, I'm not seeing it.
I'm not seeing my screen. What about now? Are you seeing it now? Are you seeing my screen now?
It's very tiny. Please enlarge the screen, please. Yes.
Okay, are you seeing it now? Pressure.
I'm not seeing it. Yes, I can see it.
So what's the slide that I'm in right now? Hello? I think you're seeing it now, right? Okay, so sorry about that. So let's continue. I was saying that cyanobacteria, they were one of the earliest type of cells that was originated on Earth. So these cells, they played a very important role. These organisms, they played a very important role in what? Synthesizing oxygen through the process of photosynthesis. So this led to a very major event called the Great Oxygenation Event, as happened around 2.5 billion years ago. Please be taking notes of these major events. Now, cyanobacteria, they are what organisms that one of the earliest organisms that had chlorophyll, which means they were able to synthesize their own food by themselves. Now, in the process of photosynthesis, they released oxygen into the atmosphere. Now, the accumulation of oxygen in the atmosphere led to what is called the Great Oxygenation Event. This happened around 2.5 billion years ago. So the evolution of complexity, that is transition from simple to multicellular organism, started from, first of all, little microorganisms, and before you know it, they were built up to form the multicellular ones which we know of today. So the next slide, we'll be looking at cell theory and structure. Any question? Fresh, hope you are still seeing my screen now.
Yeah, I can see it.
Okay, right. Do you have any question from the last slide? Okay, so no question.
Yeah, no question.
Okay, beautiful. So we'll now be looking at a very crucial topic, the cell theory and structure. So what's the cell theory? The cell theory is a fundamental theory that's kind of like guiding all life. The cell theory tells us that, first of all, every single organism on Earth, whether plants, whether animal, whether human beings or microorganisms, whether ants or elephants, all of them are made up of cells. So this places cell as principle or the smallest unit of life. So first of all, number one in cell theory is that every organism are made of cells. Two, these cells are the smallest unit of life. When you talk about a unit of something, it means the smallest part of that thing that can function as a whole. So in a whole human being, if I should slice off your skin now and look at your skin with a microscope, you notice that your skin has different compartments called cells. Now, this compartment is the smallest unit that can perform all life activity. Now, inside these cells also have structures, which I'm still going to look at. Now, inside these cells are structures. If you remove any other part from that cell, that part on its own cannot perform life processes. Now, if you bring a cell now, you are going to see the cell wall, you see the cell organelles, that's the mitochondrial, ribosome, endoplasmic reticulum, and the rest. If I should remove the endoplasmic reticulum now, that endoplasmic reticulum cannot function as a living thing. It does not have life in itself. If I should now say, okay, let me remove the nucleus on its own, that nucleus does not have the characteristics of life. It cannot function as a living thing on its own. So, but once you bring them all together, it's from that basic unit of life called a cell. So, what you're trying to establish now is that the cell is the smallest unit of life. Outside this small unit, any other further fragmentization, that is, any other further breaking down, that parts can no longer assume the full process of life. So, the three and the most important is that every single cell comes from pre-existing cells. Every single cell comes from pre-existing cells. So, now, let's use our example again. If I should bring a knife and now slice my hand or cut my hand, or peradventure I fell down and I have an injury, I will not wake up one morning and then some new cells will just appear from nowhere. No. How will these new cells form? How will that wound close up? You originate the existing cells here already will start dividing. Now, these cells will divide and what regurgitate to cover up.
Excuse, I'm not seeing your screen again. Please.
My God, are you seeing this now?
Yes, I started seeing it. Thank you.
Okay, no problem. All right. Please, hope everyone is seeing my screen. Okay, so let's continue. We are at number 3. So the 3 cell theories are: number 1, all organisms are made of cells. 2, the cells are the smallest unit of life. And 3, all cells come from pre-existing cells. All right. So let's move on to organelles and function. So when you talk about cell organelles, what do we mean? Now we know what a cell is. A cell is the smallest unit of life that can perform all biological processes. Now, what is an organelle? An organelle simply means a very small organ. That's not actually a small organ, a miniature organ. It's like an organelle is what just like a very small factory, a microfactory inside a cell that performs a specific function. Are you listening? An organelle is what a small portion of a cell that performs a specific function. So now in the cell, there are different types of organelles, which means there are different sections. Let's use an analogy of a factory. Inside a factory, there are different offices where different life processes occur. So let's use a bread factory, for example. There is probably a room where the what flour and the rest are mixed. Now, there is a baking section where the baking takes place. And then there is the office where the what cashier and the accountants take note of everything that is going on. And then also probably there may be a store inside the factory where the bread will be sold. So likewise in the cell, things are not just done scattered around in the cell. No, there is what different compartment or different life processes. So in the cell, we are going to find what's in the cell now. This picture is not clear. Well, in the cell, we are going to what find in the center a what centrally placed membrane-bounded organelle called the nucleus. So what does the nucleus do? The nucleus was it is an organelle that stores the genetic materials in form of DNA. Are we still together? So the nucleus have one major function. They store genetic materials. Also, the nucleus controls all cellular activity. So I will say now that the nucleus is what the boss of the cell. Just like in every organization, they have what the manager, the accountant, and the rest. So the nucleus is like your manager. Is what the person that tells other parts what to do and when to do them. So we can also say what the nucleus controls all the cellular activities. So let's look at what second most common organelle, which is called the mitochondria. So what is mitochondria? The mitochondria, as we all know, is what the powerhouse of the cell. So what do you mean by powerhouse? This is where energy is generated for what all the cell activity. So the mitochondria is what's responsible for energy production through ATP synthesis during cellular respiration. Don't worry, as we move on further, you'll be understanding most of these processes that occur. So the mitochondria is what? The mitochondria is what the powerhouse of the cell where what energy is produced via ATP synthesis. So let's move on to the next organelle. We have the ribosomes. Whenever you hear ribosomes, what should come to your mind is protein synthesis. Protein synthesis. So the ribosome what is responsible for production of what? Proteins. Okay, so another organelle you can also find in the cell is called the endoplasmic reticulum. The endoplasmic reticulum. Endoplasmic reticulum is concerned majorly with what? The transport and what? Synthesis. If you go further, they will teach us, I'll be teaching us that the endoplasmic reticulum has two types. Number one is a rough type, and number two is a smooth type. Okay, so let me just explain here. Just pay attention. The rough endoplasmic reticulum is called rough because its surfaces are not smooth. Very simple, right? Now, they are called rough because ribosomes are attached to them. So, the rough endoplasmic reticulum specializes in what? Synthesis and transportation of proteins. Let me come again. There are two types of endoplasmic reticulum, the rough endoplasmic reticulum and the what? Smooth endoplasmic reticulum. The rough endoplasmic reticulum are what? Concerned with protein and synthesis and transport. The smooth endoplasmic reticulum are concerned with the lipid synthesis and detoxification. And I seriously hope most of us are writing this down. So, the smooth endoplasmic reticulum is very smooth because it does not have anything attaching to its surfaces. The rough endoplasmic reticulum is rough because on its surface, ribosomes, remember, ribosome is the organelle for protein synthesis. Now, this same ribosome comes and what's attached on the surface of this what rough endoplasmic reticulum. So, you see that now the rough endoplasmic reticulum is concerned with protein synthesis and transport, whereas the smooth endoplasmic reticulum is concerned with what? Lipid synthesis and detoxification. And lastly, we are going to be looking at the what? Golgi apparatus. The Golgi apparatus. So the Golgi apparatus is what? Concerned with the function of packaging and modifying proteins. Packaging. So the Golgi apparatus, they want package proteins produced by ribosomes and rough endoplasmic reticulum. They package them, they sort them together, and then they transport them within or outside the cell. Hope you are all still together. Any question up to this point? So we're going over to the next slide. Prokaryotic and eukaryotic cells. Prokaryotic and eukaryotic cells. So, what are prokaryotes? What are prokaryotes? Karyote simply means what? Nucleus. Karyote simply means what? Nucleus. Pro means what? LS form. Pro means LS form of primitive. So prokaryotic cells are what? Cells without a nucleus. Prokaryotic cells are cells without a nucleus or membrane-bound organelles. There are what cells found in diverse environments, including the bacteria and the human gut. So that EU simply means true and karyotic means nucleus. So when we talk about eukaryotic cells, we are talking about cells that have what? True nucleus. So we now define eukaryotes as what? Organisms with cells that contains what? A nucleus and a membrane-bound organelles. So for you to differentiate between prokaryotes and eukaryotes, we are going to look at two things. First of all, do they have a nucleus? Yes or no? In prokaryotic cells, they have no nucleus. The DNA material is what? Just condensed within one part of the cell. In eukaryotic cells, they have nucleus. So the DNA material is what? Arranged inside the nucleus. Also, now look at their organelles. Their organelles, do they have what? Membranes. Membranes just like what? A covering of something. Just like your phone now has pouch, right? The phone has pouch that covers the back. Likewise, the organelles, the small factories inside the cells, they are what? Covered by what? Membrane. Okay, let me use a more common illustration that you can understand. Imagine inside your school. Now, your school is a cell. In your school, your whole entire school is what? Surrounded by a fence. You are making an assumption now. Your whole entire school is surrounded by what? A fence. Inside that school, there are different classrooms. You can have the principal office, you can have the VP's office, and you can have the lecturer staff room and then your class. Okay. So each classroom now represents an organelle. Now, let's bring your classroom now, for example. Your classroom now has what? Still what? Blocks covering it up, and then the space inside, really this space. Now, that blocks is what we call membrane bound. So, your classroom is covered by what? By a membrane. Let's make another illustration. Imagine another school now that has what? A fence. But inside that school, there are no buildings. That is, there are no classrooms. You see that school now does not have what? Membrane-bound organelles. So once you enter your school, you just stand inside the field, and then your teacher will come under the sun and start teaching you. So life processes occur without what? Specific areas. They do not occur kind of like they occur what? Inside the cell. There are no specific areas or specific places allocated for them. Hope you are still together. So prokaryotic and eukaryotic cells. Prokaryotic cells have no nucleus. Eukaryotic cells have nucleus. Prokaryotic cells do not have membrane-bound organelles. It's just like a school field without classrooms. Eukaryotic cells have membrane-bound organelles. It's just like a school field that has classrooms where different lectures take place. Also, now let's look at other things. In prokaryotes, they have circular DNA. The DNA is circular form, that is formed rounds. In eukaryotes, they have linear DNA. Linear DNA. You're that way teaching DNA is helix in nature. Helix. Okay. So now I've mentioned 3 characteristics. Number 1, no nucleus in prokaryotes. Circular DNA, no membrane bound organelle. Also, prokaryotes has smaller ribosomes, whereas eukaryotes have larger ribosomes. Okay. Lastly, examples. What are the examples of prokaryotic cells? Very common. Oncever you hear about prokaryotic cells, what should come to your mind is bacteria. So number 1 example is the bacteria. Number 2 is also the archaea. Archaea is an ancient type of bacteria. So you can also call it archaea bacteria. In eukaryotes, example of eukaryotic cells, animal cells, plant cells, fungi, and protists. Hope you are still together. Any question up to this point? Any question? Okay. So we'll be looking at the evolutionary significance. Evolutionary significance. Now, the evolutionary significance, that's the endosymbiotic theory. This explains to us that mitochondria, let me move to the next slide. So, okay, now from this slide, all of us can see this slide now. By my left hand is a prokaryotic cell, and by my right hand is a eukaryotic cell. If you look at the middle now, this middle place is called the nucleoid. This is just a region where the DNA, the circular DNA is found. On my right hand side, you see the eukaryotic, the eukaryote. Now, if you look at this cell, inside the middle part, you see the nucleus. Inside the nucleus contains the DNA, and then a region whereby the RNA is also formed. Now, okay, I was talking about evolutionary significance of these organelles, the endosymbiotic theory. Now, this theory says that the origin of mitochondria and chloroplast in eukaryotic cells originated from the free-living prokaryotic cells. Okay, so I just want you to pay attention. What I want to explain next is not in the slide. So if you have your notes, you can bring it out and write on now. So the endosymbiotic theory, this is just telling us that mitochondria and the chloroplast, which are found inside the eukaryotic cells, these are originally prokaryotic cells living on their own before they got incorporated inside the eukaryotic cells. Okay, so this helps the cells produce both food and energy, whereas the eukaryotic cell on its own provided it with protection. Hope we are still together. So let's look at mathematics in biology. Most of us that wanted to go for medical schools, we are trying to run away from biology. Without knowing that from mathematics, without knowing that in this life you cannot run away from mathematics. Okay, so we're looking at basic biostatistics. Basic is the word, basic. So everything we're looking at today is simple. So it's not something hard, and it's something you should have. So central tendencies, measures of central tendencies. So what does central tendency mean? Central tendency describes the central points or a typical value of a data set. So whenever you think of central tendency, we are trying to figure out if I should give you, okay, if I should give from number 1 now to number 100. Central tendency is trying to find the middle ground, the middle number, or the most common occurrence within these data set I've provided. So this measures now tendency tends to summarize data into a single value that can help us with understanding and comparison. So central tendency does 2 things. It helps us to what? Understand a given data set or to be able to what? Do comparison. So measures of central tendency. Number one is the mean. Number two is the median. And number three is the what? Mode. So we'll be looking at these three basic ones, and then I'll introduce us to the two advanced concepts, the weighted mean and the trend mean. So we'll not be going into that one now. So for the mean, what does mean mean? Mean simply means average. Average. So whenever you hear about mean, it's not what do you mean? No. The mean I'm talking about here is what? Average. So the mean simply means what? Arithmetic average of what? A given data set. If people are writing, let me repeat it. Mean simply means what? The arithmetic average of a given data set. So for you to calculate your mean, you would have to add up, you sum up every number in a given data set and then divide by the total number in that set. Now, for example, imagine I should give you from number 1 to number 10. That is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. If you want to calculate for the mean, you sum up the individual number 1 plus 2 plus 3 plus 4 plus 5 plus 6 plus 7 plus 8 plus 9 and plus 10. Then you will divide it by what? The mean data set. Are we together? So I would love us to do this thing now. So if you want add up all the numbers in a given data set, you now divide it by the average number, you get what? Your mean. So let's look at the next one, the median. The median simply means the middle value in a data set. The middle value in a data set. So if you want to calculate for the median, you first of all arrange a given data in ascending or either in ascending or descending order. Okay. So if I should give you 5, 6, 1, 2, 8, 9, 10, 5 to 3, 4. If I should tell you now, find for me, you have to arrange it from number 1 to number 10. That's in ascending order. Or you arrange it from number 10 to number 1. That is in descending order. When you are finished arranging it, you now what? Divide it by 2 to find the middle of that. Let's go again. If you want to find your median, you arrange either in ascending order or in descending order. Then you find the middle ground. That's the middle of that set. And then from there you pick out your what? Median. Imagine I should give you 10 numbers in random order. What you do is you arrange that number either in ascending or descending order. Then you what? Find the middle ground. Okay, so let's look at the next what? Next central tendency or the mode. This is my favorite during secondary school period. The mode simply means what? Highest occurring value. The most occurring, the highest occurring value. So the mode is also what most useful for categorical data. That's data that can be grouped. So I'll do a quick recap now on these three basic central tendencies. Number one is the mean. The mean is the arithmetic average of a data set. Number two is the median. Median simply means the middle number in a given data set after being arranged in either ascending or descending order. And the third one is the what? The mode, which is what? Most occurring data set. So the advanced concept. In the advanced concept, we have the what? Weighted mean and the what? Trimmed mean. In our next lecture, we'll be discussing on this one further. Okay. Application example. So from this data set, 12, 15, 15, 18, and 20, I would love us to calculate for our mean, median, and mode. Are we still here?
Hello, sir.
Hello, I'm hearing
Hello?
you.
Yeah, the
Hello, are you hearing
trim
me?
mean and the word mean is not well understood. Please expatiate on it more.
All right. So the weighted mean simply means what is a mean whereby each value in a data set is assigned what a weight based on its importance or frequency. Hello,
Yes, I'm listening.
are you listening to me? Okay. So
Yes,
I see
sir.
that Okay, listen carefully. Now, the weighted mean is what? Is just a mean whereby each data set is what? Assigned a weight. Hello? Is a mean whereby each data set is assigned a weight
Hello,
based
I'm
on
listening.
all right. It was a mean whereby each data set is assigned a weight based on its what? Importance or frequency. So now imagine I should give you a number. Imagine I should give you a series of number. All I'm trying to find out is what is the what weight of every given aspect of it. Now let's use this our example now, for example. 12, 15, 15, 20. If I should tell you now to find the weighted mean, I'm asking you in each of these numbers, what is what? The weight of them. Are you getting it?
Yes, I'm listening.
Okay.
The weight of it, I think it should be the addition of all. Is
No.
it?
Listen again. The weighted mean is just like what? A regular average, okay? It's just like a regular average, but it gives now what? Importance to each value. Are you still with me? The weighted mean is what?
How
It's
do I
just like
give importance to each value? What's the criteria? How do I give value to each?
now you multiply each value by what? Each weight. Okay,
By
bring your pen
this
and paper.
one.
Let's solve this now. By the
Okay.
weight. Bring your pen and paper. Let's solve this now. Hello.
Your voice is no longer audible.
Are you with me?
Yes, I
Okay,
can
so
hear
write
you
down,
now.
write down, write down these numbers. 12. Have you written it? 15. 15.
I wrote 12, 15, 15, 20.
I put 18 there.
Okay.
Okay. Now,
Okay.
in weighted mean, you are the one by yourself that will assign the weight to each number. So in this case, I'll be the one assigning a weight to them. So what do the weight mean? It simply means what? How important each number is. So if I'm to say now, okay, for a popular example is in your grading system, we have A, B, C, and D. Now, in A, you say that A is worth 5 points. You are okay, you just finished secondary school, JUPEB. Now, in the grading system we use in school is that A means 5 points. That is, A has the weight of 5 points. B has the weight of 4 points. C has the weight of 3 points. D has the weight of 2 points. E has the weight of 1 point. And F has the weight of 0 points. Are you with me? Hello? Please
Yes, I'm
respond.
with you. I'm with you.
Exactly. So whenever you're talking about weighted mean, you are now calculating the mean, but based on the importance you have assigned to all of them. So if you should come to an exam now and you get an A, I will assign to you 5 points because we are now seeing A as being more important, right? So if someone else should get a B, the weight of that B is what? 3 points. It's not equal to A. Are you with me? So now for 12, let's assign the weight of 12 to be 3 points. Now for 15, attach the weight of 15 to be 5 points. Now for 18, let's say give it
Your voice is cracking. You said 12. I should assign what point?
3
12
points to
and
12.
15 and 20.
Wait. 12 will be 3 points. Are you listening to me? 15 is 5 points. Now your 18 will be 6 points. And your 20 it will be 7 points.
$12. What point?
Yes. 12 is 3 points.
18 ńkọ́?
18 is 6 points. 20 is 7 points. Have you done it?
Yes, I've written it.
All right. So for you to calculate your weighted mean, you have to multiply each number by the weight assigned to them. So now multiply 12 by 3. Others online, you can also follow us. Now multiply 12 by 3. So multiply out and tell me when you are done.
36.
Beautiful. Do the same for 15. Are you done? I think. Are you done? So, okay, let's proceed. When you are done, just tell me. Now, when you multiply 12 by 3 points, you multiply 15 by 5 points, you multiply 18 by 6 points, you multiply 20 by 7 points. You have now what? Gotten the weight of each value. So what you do next is do what? To add up all these weights together. That's, you now add up what? 12 times by 3 is 36. So you now do what? 36 plus 5 times by 15 is 75. Since 15 appeared twice in this our example, you do it again. 15 times by 5, another 75. Now, you now add up everything. 18 times by 6, 18 times 6, 108. 20 times by 7, 140. So when you add 36 plus 75 plus 75 plus 108 plus 140, you should get around 434. 434. So now the 434 is what? The sum of the numbers and their weights. So next thing you will do is to add up all the weights together. That is what you multiplied with the 3 plus 5 plus 5 plus 6 plus 7. When you add them up together, you now what? Get the sum of the weights. That is 26. So you will now do 434 divided by 26. This will give you what? Your weighted mean. If you are in the university, this is how they calculate for your CGPA. After your exams, they will now assign to you grade points. A 5 points, B 4 points, C 3 points, D 2 points, E 1 point. So after your whole exam, some are doing 12 courses, 15 courses, they will get all of these things, calculate your what weighted mean, and give you what CGPA. Are we together now? Have your question be clear?
How do I know the weight to assign to them? Do I assume? If I assume, another student will assume, another one will assume. We will not arrive at the same thing. Huh?
Listen to me. The weighted mean will be given to you. I did not include it here because I did not intend on solving it, okay? So the weighted mean will be what? Given to you. Are you hearing me? Hello? So the weighted mean will be given to you, just like I was using your grade points as example. In every school, their grade point is what's given to them. Like my own university, they will tell you A is 5 points, B is 4 points. It is certain. So in your exam, if you should see the weighted mean, which I doubt you will see, but if you should see it, they will give you the weight of each value. Okay? Hope this has been clear. So let's move on. Now, practical microscopy. So the last one we'll be looking at is what how to use a microscope to run a very simple experiment. So for you to use a microscope, number one, you would set up your microscope. So you now place the microscope You place the microscope on what? A stable surface. You what? Place the microscope on a stable surface. You turn on your light source. When you turn on your light source, I don't know how many of us has what used the microscope here before. When you turn up your light source, you adjust the mirror for proper illumination. And you only use what? That is low power objective lens. Objective lens, you see like times 4, times 10 magnification. So we start with what? A low power one. So the next step, you place the prepared slide and adjust the focus. So now, let's imagine you want to view the ants under a microscope. First of all, you set up your microscope and choose a very low magnification. Start with around times 4, 4x. Now, you place the ant in a slide. You place the ant in a slide and what? Put it and ensure that the slide is clean. So after that, you would place it on the stage and clip it. So number 3, you observe the sample, switching magnification as what's needed. So after you have placed this prepared slide under your microscope, you now look into it using the objective lens. Now, when you look into it using the eyepiece, when you look into it, you will keep changing the lens to find the magnification that suits you. After that, you now want to observe the cell types. So let's imagine we want to observe what an animal cell. What are we likely going to see? To say, okay, this is an animal cell or this is a plant cell. So for an animal cell, first thing you should notice is what? A cell wall, not a cell membrane, a cell wall. So when you see a cell wall is what? Is a very rigid structure that would is the atmosphere structure that would surround the whole entire cell before you see the cell membrane. So this cell wall, after the cell wall, next thing you see in the plant cell is also the what? The chloroplast. The chloroplast is the what? Green coloring pigment responding for photosynthesis. After the chloroplast, the next thing you should see is what? A centrally placed large vacuole. Are we together? Now, if it's the animal cell you're observing, you notice that the animal cells do not have cell wall, and they have a small vacuole, and then a visible nucleus as well. Okay? Any question up to this point? I wish we have a microscope. We'll now run a simple setup. Any question at all? So I should move on to conclusion. So in summary, we looked at the origin and evolution of cells, the difference between prokaryotes and eukaryotic cell. And I made mention that the prokaryotic cell do not have a well-defined nucleus, and they also do not have cell organelles with double membrane. The eukaryotic cells have a true nucleus, and they have cell organelles with double membrane. And we also look at practical microscopy techniques and the application of biostatistics and biology. And we learned mean, median, and modes. And we also learned weighted mean. Okay, now by next tutorial, we're looking at more other topics. Please, do anybody have any question? In the answer of question, this is the end of the class. Okay, I'm in the chat section. Okay, you can't hear me very well. I'm so sorry. I was presenting. I could not change the chat session then. Any question? Okay, then good night, everyone.
Yes, from the calculation you did, which one is yes, which one is our weighted mean now?
Okay,
Is it
so where did you stop?
Is it 434 or 26?
That's the weighted mean.
Hello?
Where did you stop? Hello, where did you stop? Hello?
Can you hear me?
I can hear you.
I say, which one is our weighted mean now from the calculation we did? 434 when we add up the products of each, and then the addition of the weights. So which one is the weighted mean?
Now, for you to get your weighted mean, you will do 434 divided by the sum of the weights. That is 434 divided by 26. So do it quick and tell me.